Good morning, good afternoon, and good evening, everyone, and welcome to our webinar titled Improving Sleep to Slow the Progression of Alzheimer's Disease. This webinar has been sponsored by Harvard Bioscience, so a big thank you to them for helping to make this event possible. Joining us today, we're very fortunate to have Dr. Rohan Jagirdar, an instructor at Baylor College of Medicine. His presentation will discuss his research on EEG activity in a mouse model of Alzheimer's disease to determine the effects of improved sleep on disease progression. I'm Sarah McFarland from the events team here at Inside Scientific, and I'm very pleased to be your host for today's event. Rohan, thanks so much for joining us today. Take it away whenever you're ready.
Thank you, Sarah, for the great introduction, and thanks to DSI and Harvard Biosciences for providing me this amazing opportunity to present my work through this webinar series. And it's really great pleasure to present my work along with many other talented speakers in this webinar series. And I would like to thank everyone for attending my talk. I'm Rohan Jagirdar. I'm an instructor at Baylor College of Medicine, working in the lab of Dr. Jin. Today, I will be presenting my work titled Restoration of Activity in Thalamic Reticular Nucleus Improves Sleep Architecture and Reduces A-beta plaque deposition in Alzheimer's disease mice. So you'll probably know that Alzheimer's disease is associated with the learning and memory deficit. However, how AD leads to learning and memory deficit is not completely clear. We have learned a lot about the AD from the families with the familial autosomal dominant mutations, such as this shown here, which all lead to increased levels of A-beta in the brain. To study how A-beta leads to memory deficit, many transgenic mouse models have been created to express this human mutation. They also produce high levels of A-beta and develop memory deficits. So the field has learned a lot using these mice. Much of the field has really focused on the hippocampus to study memory, but we are still far from having the therapeutic strategy to treat Alzheimer's disease. We now realize that the hippocampus-centric view of Alzheimer's disease cannot really explain the entirety of this disease. Many symptoms of AD are related to the hippocampus, like memory impairment, mood disorders, and some types of seizures. However, there are other symptoms like deficit in sleep maintenance and slow wave sleep, attention deficit, and occurrence of other types of non-convulsive seizures not regulated by the hippocampus, but rather they are primarily associated with the thalamocortical network. Thalamocortical network interacts with the hippocampus and is comprised of a reciprocal communication between cortex and thalamus. The inputs from the deep layer of cortex to the thalamic relay nuclei are regulated by a major control nucleus called thalamic reticular nucleus, or we call it TRN. TRN is purely inhibitory, so it gates the propagation of sensory afferent information from the periphery to the cortex, which is particularly important during sleep. We therefore hypothesize that the TRN is a master regulator of cognitive and behavioral impairments in AD. To study role of thalamic reticular nucleus in progression of disease, we use J20 Mouse Model that expresses human APP with familial mutation linked to AD. These mice produce high levels of A-beta that accumulates to measurable level by the age of 1 month. However, formation of A-beta plaques do not start until the age of 6 months. Around the age of 2 months and 3 to 4 months, they exhibit synaptic loss and cognitive deficit. With that being said, we are really interested in what happens in this early phase so that we can try to understand early events of the disease. What we have seen here that the earliest behavioral changes in these mice, like epileptiform activity and seizures, as well as sleep fragmentation and deficit in slow-wave sleep. This all started few years back when our lab discovered that the J20 mice exhibit decrease in TRN activity, resulting in disinhibition of thalamic relay, so increase in thalamic cortical activity. Using phosph delta phosph, an activity-dependent marker, to look at active cells, we found fewer active cells in APP mice in comparison to NTG. There is no neuronal loss in TRN that's been shown here with the help of NuM. Cells are there, but they are just not as active. And when we looked at the downstream thalamic relay nuclei, we found they were disinhibited and showed more active cells in APP mice than NTG mice. So how does this affect sleep? If TRN is hypoactive, there will be no blocking of sensory information going to the cortex that would interrupt sleep. Indeed, that's what we found, that the APP mice experience more frequent interruption during sleep using video monitoring and software analysis. That shows here 50% increase in number of awake bouts and significant decrease in average length of sleep bouts. So in a brief, we are bringing TRN, sleep, and Alzheimer's disease together. Role of TRN has been reported in several studies in maintenance of and generation of activities like slow wave activity, spindles during non-REM sleep. Also, slow wave sleep is critical for memory consolidation, for clearance of waste material from brain. Some other studies reported decrease in slow wave sleep in AD patients and disruption of slow wave activity in healthy subjects increase CSF A beta level. A study that was published about 7 years ago proposed that our brain starts cleaning when we go to sleep. So I'm going to show you here one of the TED Talks from Jeff Elif. So they demonstrated that the glymphatic system clears metabolites from the brain during sleep. During in vivo multiphoton imaging, they injected green tracer in cisterna magna of the mouse brain when it's active. We can see here in vivo imaging when the mouse is awake. Look, the amount of green tracer flows through the brain, a very few. And now when a mouse goes to sleep, they inject red tracer with CSF flushes through the brain. Notably, A beta is activity dependently made. So sleep is important for two reasons. One, it reduces neuronal activity and therefore A beta production. And two, it enhances A beta clearance. Decades of clinical studies are failing to control accumulation of A beta plaques in the brain in Alzheimer patients. And we have the power of sleep to rule that. However, the field hasn't identified a specific brain region that is responsible for sleep deficit in AD patients. And we think it might be TRN. Indeed, when we looked into our own APP mice, it does look like sleep regulates A beta levels. The area covered by A beta plaques in cortex and hippocampus, as shown here in the graph, increases with the number of awakenings. This behavioral system we previously used to study sleep did not allow us classification of sleep into slow wave sleep and REM sleep. So instead, we use this amazing EEG-EMG telemetry system that is developed by Data Science International that allows us to record brain activity and muscle activity and helped us to score sleep in field moving mice. What we see here in the next slide is a screenshot of hypnogram for the period of 6 hours of light cycle. Neck muscle, EMG, EEG signal. Based on EEG and EMG, I could score active wake, quiet wake, REM, and slow wave sleep, that is a part of non-REM sleep. It depicts how the sleep looks like for an NTG control mouse. Just a reminder for everyone that the mouse sleeps during the day, unlike all of us, like a human being, that is during the light cycle. And now if we compare the sleep of NTG mouse with an APP mouse, There are more awake bars and low amount of this flat red line that indicates the slower sleep in comparison to NTG mouse. And we can clearly see that the APP mice awake double the amount of time than NTG mice, spend more time in awake duration, and significantly lower amount of time in slower sleep duration, and also decrease in total amount of sleep. So based on this observation, we hypothesized that a hypoactive TRN could lead to sleep fragmentation, reduction in slower sleep, and that would be resulting in A-beta clearance, which is ultimately have a causal effect on memory deficit. With that, naturally we came up with the question, can we just restore the activity of TRN? So we decided to use the chemogenetic technique that is also called as a DREAD system, which are basically designer receptor exclusively activated by designer drugs. We express an excitatory DREAD in TRN using an AAV2 virus, and that allows us to activate TRN neurons by systemically administering synthetic drug called clozapine N oxide. So are we able to activate TRN with the DREADs? The answer is yes. So we used, like I shown here, GAD2-CRE mice to selectively target GABAergic neurons of TRN using AAV2 viral vector, carrying HM32 mCherry expression cassette. Here it shows how nicely it targets the TRN and get a good expression of DREADs, as seen here with the help of mCherry reporter gene. These DREADs are functional only when you administer the mice with the CNO systemically. We can see here in an in vivo activation of TRN that the mice that are treated with the CNO are more phosphate expressing active cells than in cell treated mice. We also perform slice physiology to validate DREAD-mediated effects in thalamic slices. Thanks to our collaborator from UT Health, Houston, Michael Barland and Frederick Seip. He's a previous postdoc from his lab. So when he applied CNO in a BAT application, we saw nice activation of TRN. And if we recorded an downstream target, we saw an increase in inhibitory responses. In the slices from mice that were expressed only with mCherry in TRN did not exhibit activation of TRN neurons after CNO in a BAT application. Since this strategy works nicely to control TRN activity, we further crossed the mice with APP mice and studied sleep behavior in these mice. So to test whether TRN can alter sleep-wake cycles in APP mice, we designed a protocol as shown here. So APP mice were injected with the DREAD virus in TRN bilaterally at the age of 2 to 3 months. That's right when we first saw they start showing sleep fragmentation. 3 weeks later, all the mice received saline injection intraperitoneally at the start of a day cycle. That's when mice sleep. And on a day 3, a group of mice received CNO injection intraperitoneally and a group of mice received saline. And we recorded EEG and EMG signal during these times. In the next slide, I can show you the screenshot for the first 6 hours after saline treatment in an APP mouse. Now, if we compare that after CNO treatment, we can clearly see how the slower sleep, the red line is more consistent and a fewer episodes of awakenings. That means fewer interruptions during sleep. When we quantified this data, it prominently showed that APP mice with the saline have the expected increase in awakenings. But in APP mice treated with CNO, we normalized the awakening to NTG level. We see the effect strongly during the first 6 hours to some extent, and to some extent during next 6 hours. We can also see rescued increase in slow wave sleep duration in APP-CNO mice. So according to our hypothesis, increase in slow wave sleep and fewer interruption during sleep will reduce A-beta plaque load. However, with a single activation of TRN, we could only see the effect lasting for about 6 to 8 hours. So we slightly modified our protocol. Here, we injected APP mice with AAV-2 HM32 virus, but this time we used 6 months old mice just before they start showing plaque deposition in cortex and hippocampus. 3 weeks later, all mice received 1 saline injection as a baseline. Then a group of mice got a 30-day injection of either CNO or saline as a control. After saline treatment and every 5th day of the treatment, we recorded EEG and EMG using telemetry system in both groups. Here we can see the sleep shown here for the first 3 hours after saline treatment. And now if you look at the sleep after first CNO dose, as we have seen in acute treatment, there are fewer awake interruptions and more stable sleep. And now, if we look at the rest of the days of CNO treatment, the effect lasts throughout the treatment period. During first 6 hours after treatment, we can clearly see a significant decrease in number of awakening and increase in slow wave sleep in treatment group, indicated by here by bright red dots. That's throughout the treatment. We did not see any effect in REM sleep. However, we saw overall increase in total sleep in treated group of mice. So after 30 days of treatment, we were interested in how it affects chronic activation of TRN in helping clearance of A-beta plaque. So what we did, we basically collected the brains and processed for A-beta staining. We can see here that A-beta plaques stain in cortex and hippocampus of saline-treated APP mouse. And if we compare that to the CNO-treated mouse, there are a lot fewer plaques in the cortex and hippocampus. Quantification of staining shows a nice decrease in cortex and hippocampus, and both areas combined. So here on the graph shows the combined analysis of area covered by A-beta plaques. Further, we were interested in knowing if the hypoactive nature of TRN is also evident in AD patients. To basically investigate that part, we collected formaldehyde fixed postmortem specimens of anterior thalamic tissues from Neurobiobank, that is NIH-affiliated brain bank. And we processed those tissues and stained for phosph and delta phosph, the activity dependent marker. Like I have shown here, this is the control patient. On the left side, you can see TRN, and on the right side, you see ventrolateral nucleus of thalamus. Now, if we compare the staining of phosph delta phosph with MCI, it's mild cognitive impairment, and AD patient, we can clearly see in AD patient there are a lot fewer phosph delta phosph stained cells in TRN. In parentheses, what we see here indicates the Braak stage. The higher the Braak stage is the severe tauropathy. We analyze our data, and what we found that in 80 patients, postmortem samples, we've seen a significant decrease in phosph delta phosph expressing cells in TRN. And if we analyze the phosph delta phosph TRN cells and correlate with the Braak stages, we see there is a positive correlation with a Braak stage. That means that the severe the tauopathy, this significant decrease in phospho-delta-phospho cells indicates the severe tauopathy in the patients. So the next question, obviously, as we have tested in our APP mice was, so is there neuronal loss? So for that purpose, we performed histology using hematoxylin and eosin staining. The data is not shown here, but we did not see any differences. However, we were not sure whether that just stains neurons. It could also stain microglia and astrocytes. So we further went on with the neuron staining, and what we found here that there is no neuronal loss in TRN of AD patients. And if we compare the correlation analysis between phospho and delta phospho with the neuron positive TRN cells, that is, they are not correlated. So it simply says that the cells, like we have seen in our APP mice, that the TRN cells are there, but they are just not as active as in control. So with all that being said, decrease in TRN activity in APP mice gives rise to sleep fragmentation and decrease in slow wave sleep early in the disease and increase in A-beta plaque accumulation. Affected sleep patterns that are observed before memory impairment and A-beta plaque deposition could be leading cause for the progression of the disease condition and might as well in AD patients. When we activated TRN using DRAIDs, we normalized activity in TRN and improved sleep maintenance, increased slow wave sleep, and decreased A-beta plaque, A-beta accumulation. The role of sleep in AD is really hot topic right now, but until now, no studies have been able to identify a brain region that is responsible for the sleep deficit, much less find a way to selectively increase slow wave sleep. We think targeting the TRN could be a novel therapeutic strategy to improve symptoms and modify disease progression. With that, I would like to conclude my talk and thank my PI, Dr. Jeannie Chin, and the members of our lab, and all the past members that helped me in this project to perform experiments and collecting data, as well as our collaborator Michael Bayerlein, Frederick Seip, JJ, and Joseph for doing slice recording for us. And it's never complete without the great funding resources. In this case, Ruth Gebrard from Biomedical Research Foundation, they were ready to provide us initial grant that helped us to buy various equipment that were necessary for the sleep analysis in our early stage of experiments. And eventually, I can thank enough everyone and DSI and Harvard Biosciences for providing me this opportunity. And ready for the question, I'm going to hand over to Sarah now.
Okay, so our very first question here is, do APP mice spend more time sleeping in the dark phase as they exhibit sleep disturbances during the light phase?
Yeah, that's pretty interesting question. We looked into the dark period of this mice, but we did not see any changes in a sleep pattern or neither the amount of sleep they spend in a sleeping. So yeah, we did not see any changes in the dark period.
Okay, great. This next question is, does the TRN show A-beta plaque deposition?
Oh yeah, that was another interesting question. Yeah, no, and that's, I think, I don't know, should I say fortunate thing, but yeah, we did not see any change, A-beta plaque deposition we tested about 14 months old mice. So we could see only A-beta plaque deposition in cortex and a hippocampus, but nowhere closer to TRN. And we also investigated 80 specimens, postmortem samples, and we did not see any plaque deposition in ventrolateral nucleus as well as in TRN.
Okay. This next question is, did you use male or female mice in your studies?
We use both sexes. We use male and female mice.
Okay. This next question here is, did the TRN activation also restore cognitive deficits in APP mice?
Yeah, that was a really burning question, and we tried really hard to work on that part. So we tested object location memory in our mice, but unfortunately, because of the daily treatment with the CNO and also with the telemetry, it was really hard to find a conclusive results in the memory task. We are struggling on that part, how we can improve our behavior technique. At the same time, we can have the sleep recording and provide CNO treatment on a regular basis without using aversive method like injecting mice every day in the morning and then doing testing and behavioral testing in them. But we did not see, and that's our next task to do. We're gonna check especially long-term memory. In this case, after training period, we're gonna do 24 hours of gap and then do the testing in mice.
Okay, great. That seems like a really awesome next step for this research. So that's really cool.
Yeah.
The next question here is, do you know why the TRN activity is reduced in APP mice?
That's other side of the project where we are collaborating with Michael Bayerlein and some other group in MD Anderson to investigate that part where exactly TRN is the reason behind TRN having this hypofunction. But currently we have just a few answers there right now that we have tested connections between the cortex and thalamus, and as well as from basal forebrain to TRN, which basically comprise of cholinergic inputs. As yet, we did not find any conclusive data based on a cholinergic input, but we saw there is increase in muscarinic component. But however, that wasn't really significant data. But we also started looking into are there other intrinsic properties that change in TRN? So recently there was a paper from Bayerlein's group they published on a TRPM4. We are investigating on that part, but still the answer for that puzzle question is still remaining, and we are investigating on that further. Thank you.
Great. Yeah, these are great questions. Keep them coming. We've got another question here. It's kind of a long one. In J20 mice, overexpression of APP leads to accumulation of A-beta. Do you know if accumulation of A-beta in these J20 mice could be responsible for the decrease in TRN activity?
Yeah, that's another interesting question. Thank you. These are all the questions we are investigating step by step. But to coming towards whether the A-beta, so A-beta accumulation we will have to test in young mice. So for that case, we will have to study the soluble A-beta. And we will be doing that in our next step. We will be doing the microdialysis and see whether the A beta has any production and the clearance has an effect on TRN activity. But apart from that, we also try to investigate whether the overexpression of APP is leading cause for if whether it is a leading reason for TRN hypofunction. So overexpression of APP leads to A beta accumulation in J20 mice. But there is a negative control mouse line, which is basically I5 mouse line, which has a wild type overexpression of APP, which does not lead to A beta product accumulation. So we tested in those mice and we saw no change in TRN activity. And we also tested some other mouse lines like PSAPP and TG2576. And we saw in those mice, the TRN exhibited a similar hypofunction as we saw in J20 mice. So the question left whether the A beta accumulation is whether is it responsible for TRN hypofunction, but that's the part we are trying to investigate more in detail.
Great.
Thank you.
That'll be really cool. The next question here is for the chronic sleep study with chemogenetics, were animals dosed daily via injections or were animals hooked up to infusion lines?
Oh yeah, these ones were because we wanted to regulate the injection. Obviously, we can do that with the implantable, but however, we wanted to provide the bolus injection. So mice received daily injection of CNO at 7 in the morning, around right when the day their light cycle starts.
Okay. The next question here says, well, first it's a compliment, exciting data. The next part is the question. So do you have a CNO control group in the APP mice? And for the post-mortem studies, did you control for time of death?
Yeah, so thanks for the compliment. If I understood the first part of the question, the control for that's like M-cherry mice expression, right? So for the M-cherry, if I'm right, so we tested M-cherry as a control for HM30Q and provided CNO in non-transgenic mice and also in APP mice. I did not show that data here, but we did not see any changes in sleep patterns or any improval or any effect on sleep fragmentation after CNO treatment. And to the second question, so these samples we received from Neurobiobank, they were provided, we selected the samples based on their postmortem interval, so basically the time when they've been collected. And so they were ranging from about like 5 hours to 24 hours of gap. And based on, we were also interested whether like the postmortem collection of the samples would affect anything in our findings. So we basically tested whether PMI hour has any effect on the phospho delta phosphate expression, but we did not see any correlation. So yeah, we did record the PMI and obviously that came up with the samples, but we did not see any reason to have changes in phosphate delta phosphate.
Okay,
Thanks.
Great. Next question here. Is there any impact of oxidative stress on the TRN?
Oh, wow. Honestly, I don't have an answer for that. It would be interesting to study the mitochondrial dysfunction in TRN in this case. That would be a great way to look at the dysfunction in TRN activity, but we haven't really, yeah.
Mm-hmm. Maybe somebody else will pick that up for you.
Yes.
Next question here. First is a compliment. Really nice talk and very interesting results. And the question is, have you done behavioral analysis on these mice?
Thanks for the compliment. Yes, the behavioral analysis, that's the next part what we are wondering to do. First is the memory task. So our lab is equipped with mostly memory tasks, but we would like to study other behavioral paradigm in the Alzheimer mice, especially Alzheimer patient themselves, they show attention deficit. So we are interested, now we are driving our focus towards one of that answer is the attention deficit in this mice, because sleep and attention deficit go hand in hand. If you have a poor sleep, that affects your attention. So it would be interesting, and especially TRN has been studied for the role in attention deficit disorder as well as in schizophrenia. And we would like to perform some behavioral studies from that perspective. And we are kind of moving towards that direction now. But thanks for the question. That's
yeah, that'd be a great avenue for further research there.
Yes.
Another question here. Do you plan to analyze spectral activity during sleep?
So you mean overall changes in the frequency distribution during sleep? If that's the question, yes, we basically perform spectral analysis, and we saw some changes, decrease in high slow wave activity, so high delta activity, so about from 3 to 4 hertz. That was decreased in APP mice, and overall we saw some decrease in, let me check that data, decrease in alpha as well as beta activity, if I'm not wrong.
Okay.
I just need to go through that data quickly. Yeah, we saw decrease in alpha, but we did not see any like other spectral changes in the gamma frequency, especially, which is the more prime focus of the, has been a focus in Alzheimer's field. Yeah. I hope I given the right answer.
You had that data on hand.
Yeah.
Okay. So another question here. Do you know what happens at the level of TRN when you induce a time shift or the quote unquote jet lag phenomena? And can TRN activation help with jet lag and narcolepsy?
Oh, yeah. Oh, wow. Yeah, that's also a pretty interesting question. I don't know if I will be able to answer that. But TRN has been studied for its role in maintenance of sleep, and not much of a role into circadian rhythm or sleep homeostasis. But sleep maintenance itself in kind of plays a role in developing that sleep homeostasis. I honestly, I don't know much about like in a jet lag condition or in a narcolepsy, but role of sleep and A beta has been kind of studied for some extent in narcolepsy types. Perhaps it might be associated with thalamic dysfunction, or could be also because of the downstream brain control that helps to regulate sleep, which is not much of a function of TRN. Rather, it's more the maintenance of sleep. I hope I can give you more answer on that, but thanks for the question.
No, I think that was a great answer. We've got another question here. You mentioned that there was no significant change in the time spent in REM, but in that case, does it mean that REM is still normal in AD mice, signifying some sort of physiological sleep structure?
Okay, yeah, no, that's another interesting question. Actually, I don't know if the data was shown in one of the slides, but we saw variable changes in REM sleep in APP mice. So that's one of the reasons we did not see the statistical changes, statistical significant changes in APP mice in respective to REM sleep. But we saw in some mice the REM sleep was highly impaired, and in some mice it was left unimpaired. So we are investigating further on a REM sleep part whether there is any downstream regulatory regions could help improval of REM sleep in Alzheimer mice, or if there is any role of REM sleep in generation of seizures and so on with the downstream effect on the memory and A beta plaque deposition. Where we did not see statistically, but we observationally, I saw some impaired REM sleep in some mice. It's very variable.
All right. Next question here. When you activate the TRN, which neuronal systems are activated and which neurotransmitters may be released and in which areas? It's a long question.
Oh yeah, no, that's very physiological question, and that's very important question. Yes, that was the one of the first things I need to figure out how I'm going to activate TRN. So TRN, in our case, we activated whole GABAergic neurons. So TRN is, I can say it's all GABAergic neurons, and it basically provide a connection. So TRN has mainly feedback inhibitory connection to the ventrobasal complex in thalamus, and so it helps basically to control the thalamocortical activity. So in our case, we even though we activated only the GABAergic neurons that helped more the inhibition of ventrobasal complex, but in many cases, in many studies, they have preferred to activate parvalbumin neurons, which basically comprise some majority of GABAergic neurons in TRN, and then comes the somatostatin. We basically we did not activate subtypes of neurons, but mainly the GABAergic neurons. I hope that's the complete answer for your question.
Yeah, that's great.
Thank you.
Okay, we've got another question here. Is there specific biomarkers found in cerebral spinal fluid after TRN modulation?
Oh, wow. You're kind of giving me more brainstorming ideas. But no, we have not really studied the cerebrospinal fluid after TRN activation. That would be interesting to study, especially because sleep in Alzheimer's has become a really booming topic, especially because of recent finding of the glymphatic system, which so in a glymphatic system works in a way that during sleep our brain starts clearing up, resulting in expelling of any metabolites, including in this case tau and extracellular A beta. And it would be interesting to study after activation of TR and how the glymphatic system is working and the flow of the metabolites and how the clearing of the brain works. We haven't really done that, and we are not really equipped to do that, but it would be an interesting aspect to study to confirm how the glymphatic system is working when we are activating TRN. That's a great question. Thank you.
Mm-hmm. That would be a really cool study, I think.
Yeah.
Okay, next question here. Did you see sex effects in activity measure, in sleep or other measures?
Yeah, we kind of known about that. So sleep impairment is more like dominant in male mice than in even in a patients as well. It's among the male patients there is more sleep disturbances than in female patients in women. Um, in our case, yeah, I really did not demonstrate that, but we saw more sleep fragmentation among the male mice. Um, Female mice were tend to more towards the bottom of the graph. And in the cases of A beta clearance, A beta plaque deposition, clearance of A beta plaque deposition, we saw more effect in female mice than in male mice. And that was very intriguing part of the study, although we did not really elaborate on that part. But that's an interesting point. Thanks for that question.
Great. Another question here. So for the fragmentation analysis, did you have any minimum wake duration criteria to define a bout of sleep? Like if awakening duration should be at least 2 seconds to be considered awake?
Yeah, no, that's very important because especially in the cases where the muscle atonia can be counted as awake, which is especially you see them after REM period. Yeah, so we at least 10 to 15 seconds of awake period can be counted as awake. Otherwise, we did not count it as awake period. It would be false. Thanks.
Okay, great. Someone else has asked, beyond sleep, have you also checked for the effect of TRN activation on epileptiform activity in APP mice?
Yeah, no, that's another great question, and that's one of our interesting, like the part of the Alzheimer that we are really deeply interested in. So we tested epileptiform activity. We found in this case, so Alzheimer mice, J20 mice especially, they show not just J20 mice, but also other mouse models of Alzheimer's. They exhibit epileptiform activity, starting from more dominantly observed during REM sleep, and then move on to more during non-REM sleep, density-wise. And we tested in our mice after TRN activation. We did not see any changes based on number of epileptiform activity. However, that wasn't complete information. We will be more interested now to look into the spike wave discharges, which is more common phenomenon associated with thalamocortical dysfunction. And we are moving towards more specifically looking at the spike wave discharges in this mice and see if the TRN activation has any effect on that. But yeah, that's a great question. Thank you for that.
Great. And then I think just based on time, we're going to make this our last question. This question is, have you observed differences in sleep spindle parameters after TRN activation as compared to no stimulation or saline?
Yeah, this is this part I was supposed to do it at the first in hand. So because spindles are basically regulated by thalamocortical, especially thalamic reticular activity. There was, however, one obstacle in between. With our methodologies, we could not really count the spindles. So short answer is we haven't really done that yet successfully, because one of the reasons is spindle activity can be mismarked the epileptiform activity can be mismarked as spindles in APP mice, and that's the problem we are trying to solve in this case, so we can have a proper quantification of spindles in our mice, so that we can come to a conclusion that this, sleep spindles are changed. But that's the thing we really are stressing on to finding out right now. Thanks.
Okay, great. Well, I just wanted to thank you again so much, Rohan, for all of your fantastic insights today. It was really a pleasure to have you with us.
No, thank you. Thanks, Lisa, and thank you for such a great attendance and giving showing interest in my topic and our work. Thank you for the great questions as well.
Yeah, we had a really fantastic Q&A. So I also want to thank the audience for attending today. Really great questions. In closing, I wanted to thank everyone again for taking part in this Inside Scientific webinar sponsored by Harvard Bioscience and DSI. And we look forward to having you with us all again soon. Thanks.